in Dubai. I started my forensic science career in 1996 from Bahrain Police Department, Forensic Science Laboratory and Crime Scene Investigation Unit. Till 2012, I worked over there. Then I moved to Abu Dhabi Police Department. And I worked in Abu Dhabi Police Department DNA lab till 2019. Now, actually, I'm working as an academic institution called Amity University. I'm teaching forensic science in Amity University, Dubai. And I'm thankful to all the organizers of Forensic.India for inviting me to deliver this lecture once again. So let's start. The topic is crime scene investigation and forensic DNA typing. Crime is a global reality and increased today in complicated nature. Criminals executing the crimes with the latest technologies. The demand of new forensic system is crucial for the prevention and detection of such crimes. Scientific investigators must be competent enough to crack down such crimes by using latest technology. So the crimes are, changed, not like before. The modus operandi is changed because of the digital, advancement and technological revolution. The criminals are using the latest technology. So you can, or any criminals can operate in any crime if he is sitting in another country and he can execute in another country. So the opportunities of criminals are more. To prevent or detect such crimes, we need a quality forensic science service. And why do we need a quality forensic science service? That is also very important. Today, the judiciary depend high reliance on scientific evidence. Quality forensic science service can safeguard against miscarriages of justice. The forensic evidence, although secondary in nature in Indian Evidence Act or Indian criminal justice system, but it corroborates primary evidence, prove more reliable, reproducible, and neutral in nature. So it's a fact. The scientific evidence is a fact, not like eyewitness testimony or other kind of evidences. The forensic evidence which speak the truth if properly processed. The changing face of scientific investigation. In developed countries, law enforcement agencies adopting new forensic technology for the detection and prevention of the crime. Indian investigation agencies also adopted new technology in criminal investigations. In many states, I have seen they are now started adopting new technologies. But in certain extent, the quality of our forensic science system or services are not developed as international standards. Not talking about all states or in a federal system, but it needs more improvement. So crime scene to court. In any forensic evidence which you found in the crime scene, then if it is properly processed from crime scene to court, crime scene, forensic lab, then to court, until the judgment. So the quality, we have to, who will maintain the quality? All the stakeholders who are participating in the this evidence chain of custody or in the crime scene or the forensic lab. So they have to protect and secure the evidence. So then only the conviction will happen. So many a times the conviction which is not happening in the court of law because the evidence is not properly presented or analyzed properly. So we have to start our evidence processing from the crime scene itself. So crime scene evidence identification is very important. Then collection of evidence is also crucial. And submission, collection, transport, packaging, all is very important. Submission to a forensic lab. So the forensic lab is analyzing the sample which they received, comparison or comparing with the reference standards or suspects, then they make a report. And this report has to go to the court. The court So cross-examination will be there. The evidence expert testimony is there. Then the defense lawyers may argue, because nowadays the defense lawyers, they argue or they cross-examine the expert or scientific analyst, because they also getting a lot of information about forensic science. So finally, the judgment which is come from the judge, and it is depends entirely on the quality of the forensic process. So where it starts from the crime scene investigation. Lockhart's exchange principle, all of you know about it. The perpetrator of a crime will bring something into the crime scene and leave something from the scene. So that is the key of the forensic investigation. It is even more critical today to ensure that the evidence is protected and gathered in correct manner, maintaining its integrity for the proper court trial. So it's very important, maintaining its integrity, maintaining the evidence integrity is very important. Correct scene management incorporates good practice with modern equipment and quality control. If a crime occurs, the scene management is very important. And good practice, there is good laboratory practice is there, standard operating procedures are in place. The same like the good laboratory same like good crime scene investigation practices should be there. So the scene management, so it's a teamwork, not only one person is doing, it's a scene management. There are a police officer, crime scene investigation expert, district attorney or the prosecutor, a judge also may present in some of the serious crime, a forensic examiner, that means a medico-legal doctor, a detective, a specialist, for example, you can say a CBRN specialist or a ballistic expert. So there are certain specialists also. It's a teamwork. So from the crime scene, what's the first thing to do at a crime scene? Secure and isolate crime scene. Very important. The first responder to the crime scene, he has to secure and isolate the crime scene. Then document the scene. So documentation is very important because the lawyers not coming to the scene of crime. So we have to submit proper documentation to the court. Conduct systematic search for evidence. So systematic search, it depends upon each law enforcement agencies or forensic department policies, how they are doing the systematic search. Collect and package physical evidence. Very important. So how you are collecting the samples, how you are packing the samples, and maintaining chain of custody. The sequential order of the evidence collection also very important. So it starts from the crime scene. So if the crime scene sample is contaminated, so what is happening? All the entire process is invalid. So the crime scene protection, documentation, collection, evidence, which have to have a quality system in place. So secure and isolate the crime scene. Responsibility of first officer on scene takes steps to preserve and protect the scene. So the first officer, when he arrives to the scene, he has to protect and preserve the scene. And first priority, the medical assistance if required. So he has to provide that. Exclude all unauthorized personnel from the crime scene. So he is a uniformed or a police officer or a responsible personnel. He can guide or advise the other people not go to the crime scene. He has to do his own way of the protecting the scene, and established a control and exit point. So the crime scene officer or the first responder, he has to establish a common entry and exit points. So he can use ropes, tapes, barricades, and guards strategically placed. Scene preservation, very important to manage who and what goes in and out of a scene of crime to ensure that high level integrity maintained throughout the examination phase. So scene preservation is very important. So if anybody is going inside to the crime scene, he must have an visitor logbook or a visitor log. So who entered, at what time entered, what was the purpose of entering to the scene. So he has to document all these things. And crime scene mapping. It's very important. In the older days, we doesn't have much technologies. Today, we have enough technologies with us. So the crime scene mapping, if it is an outdoor crime scene, we can use the drone with attached with a camera, and we can remotely monitor all the evidence without going to the for example, sometime there is a bomb explosion scene, or so we no need to go to the scene. With the far distance you can operate or examine or map the scene. And another very important equipment or the tool which is using in the crime scene nowadays, which is a 360-degree camera. So you will get a good panoramic 360 view of it. So that is a good tool to document it. And it is very useful for the 360-degree crime scene reconstruction also. You are getting a very good, wide image. And crime scene search. So very important how you are planning to go. So as a team, you have to have a team leader. So then he guide you what to do. One can collect the evidence, one can document the evidence, one can take the photographs. So it depends, each duty assigned to each personnel in the team. And it should be, all of you know that this is the way here all we operate when we are going to the crime scene. We completely, protected with the PPE. So because to avoid contamination, first thing for our safety, then second thing from our samples there is possibility to transfer our sample to the crime scene. Vice versa also can happen. So we do double gloves technique. If we touch one evidence, for example, a bullet cartridge is there when we picked up. So with the gloves, that gloves will be removed. The second gloves we use it for that. So frequently change the gloves. So that is very important. And crime scene search, the different methods are there. So it depends upon the crime scene management policies. And instead of using the paper techniques, the older days, or now still many the law enforcement agencies are using the paper documentation. Instead of that, now there are a lot of new tools available like CSI management system and software. It's a time of technology transition. So the technology transition is happening in everywhere. So the forensic also has to adopt that technologies. Now we have the crime scene management or crime scene pads are available, whether it is in iPad or any other gadgets. So we can install a good program there. So, and it is connected to the cloud and, or internal server that can, the DGP of a state can monitor what is happening, or the IG of the district, he can monitor what is happening in the scene. So the document is sharing. It's you can share to the remote places. So instead of physically carrying the documents there, so it is very much possible with this today's technology. Now there is a video is there. I'll try to play that one. So new phase of crime scene search and documentation. There is an instrument called Forensic. So SceneSafe actually is the European distributor, but in India, Kivitsoft Chennai is the distributor. They have all these devices. So it's a very useful instrumentation which is attached with the camera with spectral filters. So this forensicoscope is a very useful instrument to use in the crime scene investigation and for the documentation for the body fluids, particularly the latent body fluids like seminal stains, because it has the range of UV to infrared, spectral variations are there, so it can properly document it. And when it is submitting to the court, this particular seminal stains which is found on the car or in the car seat, so you can link with this evidence with documentation. So it's a very good device. So no, that's the reason. If you write with the pen and paper in draw, that it will not help with the black and white or any other camera. You cannot document the stains, especially the saliva, seminal stains, or any other body fluids, the latent blood stains. You cannot document it properly. So in that case, this kind of equipments and tools will be very much useful. The other thing is very important is scene preservation. So when I have seen that many the crime scene officers, which they are moving, the police officers moving to the crime scene. So on and they are stepping on the very crucial importance blood stain evidence and all. To avoid that, there is stepping plates, which we normally use here, the stepping plates, and we walk on it to not to disturb the evidence and collect. And you can see the image. We collect the evidence, which our two legs on that stepping plates. So why? Because on that there will be another crucial evidence. Footprints will be there, so we do not want to damage it. So we use this kind of technology. And evidence marking. So the preservation covers is it so evidence marking is very important. So we use different color codes for different, for example, blood. We use the red flags, or for the body fluids, kind of saliva, semen. We use different colors, and numbering also very, and with scale and without scale normally we use. And collect and package physical evidence. The physical evidence must be handled and processed in a way that prevents any change from the time it is removed to the time it arrives to the lab. Very important. So it should be proper. The integrity of the evidence is very important when from the collection point when it reaches to the lab. So what are some sources of contamination? The breakage. So if any liquid body fluids which is collecting, so there will be a possibility for breakage and evaporation, accidental scratching, bending, loss through improper, careless packaging. So it can happen because from in country like India, you know, so we have a very big country, and we have states like Madhya Pradesh, Uttar Pradesh. It's a huge states, and so transporting, maybe the lab is located in the capital city from another urban area or rural area. The sample is carrying from transporting from there. There are chances of breakage, evaporation, and accidental scratching, all kind of this possibilities are. So the evidence integrity is very important. So how to avoid that one? So we have to have a proper packaging system, and especially even biological evidence. For example, in this image you can see a trouser. So you have seen blood stains, and how you pack it, very important. So directly, if you fold and pack it, it will not help because it will affect the DNA results. Maybe the victims and suspects' blood are there. So in the one leg, maybe the victim's blood, and you can see in the other, the top leg, there is less blood. So it could be from a suspect. So how you pack it, very important. First of all, you have to air dry it. Then you have to keep a polythene sheet or any contaminant free paper in between that. Then only you can fold, because if you fold without a cross paper or a polythene sheet, so what will happen? Both evidence the blood stains will contaminate each other, and you definitely will get a mixture profile at the end. So in order to avoid that, you have to keep a paper sheet in between and pack it in a paper bag, not in a polythene bag. Why? The polythene bag, if you are packing in a polythene or plastic bag, what will happen? The moisture will be there, and what will happen? Then the DNA will degrade. The fungus and the bacteria will grow. The DNA strand will be degraded. So in order to avoid that, the proper packaging system should be there. And actually it is provided by the law enforcement agencies or the forensic science departments, because each evidence packaging which have the chain of custody documentation on it, so we have to write it on it. But generally, when I have seen, whenever I visited India and through the media and also, so they pack, they will buy a plastic bag from a nearby grocery or a supermarket and pack it. It is not like that. So it should be a contaminant free, this bag. It should be stored separately with the forensic department. And evidence packaging, each object. So for example, the revolver or the gun, the packaging should be separate one. The knife packaging there should be a separate one. The evidence bags should be, for each type of evidence, there will be different types of evidence bags are provided by the law enforcement or the forensic science departments. And another important thing is that the packaging of liquid blood. So you have to use an additional outer crush-proof container to use to transport or the liquid blood samples or any other body fluids, and it should be packed. Then only you have to keep this the purple top this vial to this crush-proof container. Then you have to pack in a polythene bag and fill all the chain of custody and documentation and transfer to the lab. Why I'm stressing more about the contamination? Contamination is very important. If you are taking the packaging material or any consumables from any shop or any nearby places, there are high possibility of contamination. So you might heard about this strange case of phantom killer, which has happened in Germany. Every time the forensic analysts processing their sample, they will get a female DNA, and the government spend a lot of money. Whoa, this is an unknown profile which is not matching with the database of the criminal offender database or the suspect database. So they spread the investigation to the neighboring countries like Austria and France. But still, this unknown lady, killer is not, identified. Later, they found that, and she was actually working in a swab factory, the swab making factory. So her DNA transferred to while she was making the swabs. So to the swabs, and all this years or many cases they are using the swabs, and which is contaminated. So it actually, where from it came, it is from a consumables. So same like that, the packaging, the consumables are very important. And now there is a forensic standard, ISO 18385. And the consumable standards are in place. So any forensic DNA lab, they have to use, any accredited lab, they have to use this kind of consumables. So what is the true cost of a contaminated evidence? Not guilty. So DNA scientists cannot do anything. It actually happened from the crime scene. They use the contaminated consumables. So then not guilty, that means a serial rapist, he will be in the street and again repeating his crimes. So to prevent that, avoid the contamination, we must have a proper protocol, proper quality control measures. So quality assurance is very important in crime scene investigation. Protecting the crime scenes and valuable evidence during early stages of investigation is vital to ensure successful judicial outcomes. Very important. Collecting the evidence with minimum of contamination will provide robust samples for subsequent examination and forensic analysis. Transport. Maintaining the evidential chain of integrity when moving evidence from crime scene to an examination area is key in police investigation. Identify whether at scene or at a detailed examination, getting results rapidly can add substantial value to any investigation. So standards, quality evidence collection consumables, very important. Forensic DNA grade ISO 18385, DNA denatured, ISO 17025 compliant, meets FFM LM guidelines, drug-free, alcohol-free, gunshot residue-free, clean room manufactured. All this should be followed. So evidence recovery system, sexual assault case very important. It's all the swabs and the consumers must be contaminant free. So either speculum kit, vaginal swab kit, protoscope kit, or anal swab kit, whatever it is, it should be contaminant free. And lab to field. Nowadays, what is happening? The more crimes is happening, and lab to field. Now the lab is going to the field, and there is a new equipment like rapid DNA technologies are available. You know, the evidence can be processed in the field itself. So we must have very good mobile forensic crime scene investigation or unit or mobile forensic labs. It will really help in the future because of the advancement of the new technologies like rapid hit DNA. And we need to have ISO certification for the high quality standard crime scene investigation. Why do we need high quality crime scene investigation with ISO 17020 standards? Because you can see from this image, which I have got it from the newspaper, online media. So see, there are many ways to contaminate the scene. So particularly the officer is not wearing gloves, not wearing any PPE, and contaminating. And this here you can see he himself actually is standing on the blood stains, so he can transfer. So it will not be allowed in a proper crime scene investigation. And even they do like that, the staff elimination data also should be collected. So who are attending or coming in the crime scene or near to the crime scene, they are all, the sample should be collected for the elimination data, because they might contribute their DNA to the crime scene or the evidence. So the staff elimination data is very important. Anybody is visiting the DNA lab is very important. Their elimination data is very important. Even any other office boys or clerks, peons, or those who are coming in the lab, their DNA should be collected, because the contamination possibilities are very high in crime scene. No accredited crime scene investigation, ISO IEC 17020 is very important. An increasing number of countries are adopting the ISO IEC 17020 international standard as standard for accrediting crime scenes. The reason for this preference 17020 over 1025 is that a lot of professional judgment is applied in the investigation of crime scenes, unlike the performance of analytical forensic testing and calibration in controlled laboratory condition, which will covered by 17025. Nowadays, many forensic lab is 17025 accredited, but the crime scene investigation is not accredited. There is no proper standard operating procedure for 17020 in many countries, because it depends upon the investigation of officer hypothesis. He decide what to collect, what not to collect. But if there is a procedure, standard procedure is there. So even any new staff is joining to the team, he can also follow that standard operating procedure. But many countries, unfortunately, doesn't have this. Several comprehensive and clear guides on implementing 17020 in crime scene investigation have been issued by accrediting bodies in recent years. And CSI standardization, establish regulations, standards, procedures, and protocols for the proper collection, retention, preservation, cataloging, and retrieval of evidence. The evidence is the important thing. So the forensic science lab or the analyst, what he's doing, he's applying his physical, chemical, biological knowledge to this evidence, or applying all the applications to there. So then he's getting a results for the comparison purpose. Develop statewide standardized operating procedures to the storage, cataloging, and retrieval of biological evidence. Establish guidelines for an annual audit and inventories. So there should be an annual audit or internal audit will be there, then only the system will improve. Develop a tracking system for biological evidence and it is associated data from the time of collection to the disposition of the evidence. Explore evolving technologies that can improve existing evidence and custodial process. So evidence submission. Then after the evidence properly collected from the crime scene, it should be submitted to the evidence submission to the forensic lab. And then there will be a laboratory information system or LIMS in place in many labs. It's a barcoded sample, so the person or the staff receiving the sample, the sample, it doesn't know much detail. So why? Because impartiality and independence, it is there. If the name is there, and maybe he can bias, or he can do some kind of help to say maybe his friend is involved in a crime scene, so he can help him. So to avoid that, there will be a barcoded system is there. It is really helpful. Demonstrable independence may be strengthened in the investigation work, and impartiality and objectivity. And storage facility. After the sample is received by the forensic lab, the storage facility should be proper. So because I'm a member also for the property and evidence management system in the US, and there will be proper guidelines for the storage facility, how you store the samples. Immediately, if you are not going to analyze how the biological samples, whether it is in the 4-degree, or if in the -20 or -80 or the normal room temperature, there are certain guidelines for that. The storage is very important. Now, just after the crime scene, we are coming to the sample submitted to the lab and stored properly. Then the case is distributed to a forensic DNA expert, and here he is going to start his sample processing or analysis. So we will talk now about the type of DNA evidence. All of you know that the DNA is present on all our cells, which can be get from hair, teeth, bone, saliva, blood, skin cells, semen, and other cigarette butts, and touch DNA from any samples or any watches or the key. And sources of biological evidence, as I mentioned, so except RBC, wherever WBC is there, so you will get DNA. Other possible items for DNA testing: cigarette butts, gloves, bandanas, ski masks, baseball caps, general clothing, condoms inside and outside, stains on furniture, pillows, sheets, hair clips, lipsticks, letters, envelopes, stamps, plant and animal sources of evidence. So again, the Lockhart section principle, especially in the touch DNA, anytime there is a contact between two, there will be mutual exchange of matter across the contact boundary. So the touch DNA everywhere now in the COVID time also, that's why the persistence of epithelial cells in plastic, wood, any stainless steel, the persistence is there more than 1 or 2, 3 days. And transfer of DNA evidence, Lockhart's theory: biological fluids, saliva, semen, blood, epithelial skin cells, touch DNA, fingerprints. You can recover DNA from fingerprints as well. Primary versus secondary transfer. You can transfer, for example, I'm giving a shake hand to a person, and then he is going to touch another sample. That is a secondary transfer of my DNA to another place. How much sample do you need to process it required for DNA analysis? Earlier, when I started my career, I started with RFLP in 1996. Then now, actually, a pinpoint of biological fluid or any blood or saliva or seminal stain is enough to process DNA because of the power of PCR. What is touch DNA? Usually, visible stain. Usually very small amounts of DNA are deposited. Significant increase in number of touch DNA cases in last 2 years, especially property crimes, threatening letters, robbery, homicide, assault, sexual assault, and vandalism cases. So we can recover good DNA from gloves, knife handles, weapon handles, firearm grips, plastic handles, steering wheels, rope, shoelaces, electrical cords, etc. Touch DNA cases, especially in the arms examinations, we can swab. So from the cartridge or the firearm grips, you can see in this image. You can see in this robber actually, from his cap, you can get touch DNA from he's touching this on this desk, so you can get it, and the gun you can get the DNA. DNA profiles from weapons and the DNA database. The weapon must be associated with a crime seized and surrendered. The weapon cannot be seized from the suspect person or property, cannot use possession sample as an alternate way to get suspect known. Collection of touch DNA from evidence at the crime scene by law enforcement, doorknobs, counters, windows, forensic laboratory by analysts. In other testing needed, where areas of contact, grips, slide, trigger, magazine, cartridge cases, fired, unfired, any touched object. But be cautious regarding objects accessible to the general public. So there will be more chances of contamination. Touch DNA should be analyzed very carefully. Collection of touch DNA evidence, contamination is significant possibility. Impact of contamination is false exclusion of suspect or artificial mixtures. How to minimize? Gloves, masks, disposable instruments, procedure, not talking over the evidence, identification of contamination, know the DNA profiles of first responders, major crime squads, and laboratory personnel. This is what I said. The elimination data is very important. So first of all, we have to eliminate. If I do DNA analysis, I have to eliminate my DNA first, because I'm analyzing, there are high possibility of transferring my DNA sample to the evidence sample. Let's go to the process, forensic biology and DNA. Evidence collection, documentation is the first process, then evidence screening. Then you have to go to the, after the evidence screening, there is a high possibility of the positive, the presence of biological fluids. Then you have to go to the extraction, extract the DNA. Then you have to quantitate the DNA, how much DNA is present in the sample. Then from the quantitation result, you have to select which PCR kits or amplification you are going to use, whether you are going to use YSTR or the autosomal STR or the mitochondrial DNA. So which kit you want to use, you have to decide after the quantitation. Then from there, you have to go to the electrophoresis, capillary electrophoresis. Then you will, after that, you will get a genotype of that evidence sample or this reference sample. Then you have to interpret it there, because after you are getting the genotype, after the gene mapper software is there, then you will get a data. So that data is not clean data. Maybe there will be a lot of artifacts. You have to clear it out. There will be mixture samples are there. So you have to make sure properly interpretation. Interpretation is a big challenge, because most of the time the DNA samples are contaminated with many other factors. And based upon your interpretation, you are creating a report, and that report is submitted to the court. And the court, there will be a test. You have to do the testimony in the court. DNA extraction. So DNA analysis begins with DNA extraction from the biological evidence. So it's a chemical process by which DNA released from the cells. So all of you know that. So the cell, which is actually DNA in the nucleus, and you have to remove all other cellular components and other the factors. So, and taking out the clear, pure DNA from there. Then this is the extraction workflow. Dissolve the stain, cell lysis, the proteins which is coated by the protein, it is digested, then DNA separated, and DNA is, then you have to purify the DNA and elute the DNA, which we can get a good yield of DNA. Yield extraction depends on many factors, which depends upon count, white blood cells in blood and blood stains, sperm cells and sperm stains, hair tissue, bone, teeth, epithelial cells in samples such as buccal swab, saliva stains, urine, feces stains. And quality of sample stain is depend upon the stain substrate. Where is the stain? Which is on a cotton cloth, which is on a plastic bag, which is on a wooden door. So all affect the yield of the DNA. And mainly the extraction method efficiency. Nowadays, we have very good technologies are there, and the instrumentations are there. Not like before. Earlier, we were using the phenol chloroform method. It was a completely manual method, and one of the thing is that it is hazardous. Then the second thing is which a lot of tube manipulations are there, so there are more possibility of contamination are there. And there are a lot of extraction challenges. The contamination is to introduce foreign material to a sample during collection, when performing sample analysis, if stored improperly or exposed unnecessarily. Contamination may result in the presence of a DNA contributor not originally present in the sample. So this is what is happening. So a DNA contributor not originally present in the sample. Lack of partial results due to the introduction of an inhibitor or a degrading agent. And type of contamination, when analyzing the extract or doing the extraction sample to sample, because 5 cases, and in one batch processing, there are different tubes are there. So the different samples are there. There will be chances of sample to sample contamination. One sample is contaminated in one case, and the 4 or other samples will be, or the 4 case samples will be contaminated. And analyst to sample, if I'm the analyst, I can contaminate the sample. If I'm not wearing proper PPE, or if I'm touching one tube of a case, one sample of a case, and without cleaning my hand, if I'm touching the other sample, there will be a chance of contamination. If I'm using the pipette, the same pipette for all the sample, without changing the tip, so there will be chances of contamination. And sample to analyst to reagent, so there are high possibility of contamination, and that's why the DNA extraction has a lot of challenges. And another challenge is degradation. Process in which DNA broken into small fragments. What is degradation? Which will be broken into small fragments. Degraded DNA, often encountered due to the nature of evidence sample, negatively impacts the ability to obtain successful DNA result. If it is degraded, we will not get a good result. So degraded DNA cannot put back together. So, but it can be prevented. So degradation after detection, improper collection, storage can cause fungus, bacteria to grow. So I mentioned that if you pack the samples in a biological or samples in a polythene bag, there are chances of growing the bacteria is there. Excessive heat, chemical exposure, bleach, detergents, UV light. Labs may expose to work UV light as a cleaning and sterilization protocols. Internal and external nucleuses, there are a lot of chances of degradation. And inhibitors also there. So molecules or substances that can interfere with the DNA amplification. Sources include denim, indigo dye. Most of the time when you do the denim genes, denim, which is a dye which is used in the genes, sometimes it will inhibit the DNA amplification. Leather, the blood stains which is on a leather which will inhibit because of the tannic acid for the tanning process which they are using. So soil, heavy metals, and humic acid. For example, the DNA samples or blood stain which is from the farm where there is a lot of bacteria and virus in the soil, because they use the manures and all, fertilizers and all. So there will be chances of inhibition of samples. And plant products, there are a lot of nucleases are present there. So there is chances of inhibition is there. And another inhibitors, while in the extraction chemicals, there are many inhibitors are there, anticoagulants, glove powder. This is also another laboratory consumables like colored PCR tubes, which will inhibit. Plant and food products, pollen, cellulose, plant polysaccharides, all will inhibit the DNA sample. Biological inhibitors, hemoglobin, heme, semen, spermine, spermidine, hair and skin tissue, calcium, melanin, and bone and tooth, calcium, urine, urea, all will inhibit. These are the biological inhibitors. So how to minimize is very important. Follow all anti-contamination guidelines. Does your forensic lab have a proper anti-contamination guidelines or good laboratory practice there? Follow good laboratory practice. Collect the sample or non-inhibiting substrates. Minimize the amount of substrate incorporated in extraction. Try to minimize the substrate, which is the blood stain, for example, which is on a cloth. So try to minimize, only take that blood stain. Don't cut this all together and put it in the extraction wire. Modify protocols, extra wash steps. So additional wash step is there, you can use. Now after the extraction, you have to come to the quantitation. Quantitation shows how much DNA is available for amplification. DNA from extraction may be uncovered or be concentrated or diluted before the amplification. Commercial amplification kits are designed to give best result with the narrow DNA quantity range 0.5 nanogram to 2 nanogram. Kit components are optimized for a certain amount of input DNA. Too much or too little DNA adversely affect the DNA interpretation. So quantitation, why we do quantitation? To how much DNA which is required, which is to optimize the DNA quantity for a particular commercial PCR kit. And may determine if other amplification methods are appropriate for testing. Mini STR testing if DNA quantity is small. So after the quantitation, we found that there is not much DNA and the sample is degraded, and then we can immediately select the mini STR, because if you go for identifier or identifier plus or global file or any other kit, we will not get a good result. Can be used to troubleshoot data, can be used to check optimal amount of DNA for new assays. Some quantitation methods can target specific types of DNA, Y or mitochondrial testing. Then after that, we are going to the PCR amplification. After DNA extraction, DNA is amplified using the polymerase chain reaction. PCR amplification makes many copies of the DNA to allow the detection. I think that this is from a small single strand, we can make million copies of DNA. And after that, the PCR, we are subjecting the samples to the capillary electrophoresis. All of you know that. So this is a fill the polymer with the solid. This is a capillary. Here the inlet cathode and outlet anode. There will be an argon laser light. So in this capillary, there is the burn capillary. Then from here only the light will pass to that detector, and all the PCR samples is dye labeled, so there will be colors will excite the light will excite the dyes, and the dyes will with the fluorescence, the light will get the reflect back, and the camera will detect, and you'll get a color profiles or a data. So DNA analysis, short tandem repeats are short stretches of DNA that are repeated several times at a particular location on a chromosome. The number of repeats and therefore the length of short tandem repeats varies from person to person. So that is the individuality or the power of the DNA. So each individual has separate DNA sequences, except identical twins. All right. So I don't want to spend more time on this one now. At a single STR locus, you can see here CAG 5 times repeated, and here CAG repeated 6 times. As this is the father, he has a 5 and 6 alleles. Here you can see the STR is 4 and 7. You can see the baby, the 4 and 6. But other possibilities are 4 and 5, 5 and 7, 6 or 7. These are the other possibilities at a single locus. So generally, we do 15 locus. No, it's a single locus. Actually, this is the possibility. So the if you add more locus, then the discrimination power will be very higher. So this is the STR identification pathway. So this is the crime scene evidence sample. Then the DNA lab. So the evidence data, the 10 and 12. So which infer the evidence genotype is 10 and 12, which is compared to the non-genotype. If there is a suspect is there, or a reference sample is there, or a database is there, so you can compare with 10 and 12, which is matching. That means the sample is matching. Then, one second. Now you can see here the DNA profile. This is the question stain, and the second one is the suspect 1 and suspect 2, which is matching with the question stain. The suspect 1 is matching because all because all the alleles, which is 16-16 and 15-16, and it is one loci. So if you have more locus is there, so you can see more discrimination power. So here in this case, you can see that the first one was a clear profile, DNA profile. The second is a mixture sample. Is it possible to compare and this samples? It's very difficult because it's a mixture sample. It's very difficult to compare. It could be from the contamination from the crime scene. Maybe it's a sexual assault case, male, female DNA involved. So it is very difficult to compare. So the evidence data in a contaminated sample, it will be like this: 10, 11, the 3 alleles you can see here, and it's the possible genotypes. will be 10, 10, 10, 11, 10, 12, 11, 11. These are the possible genotypes. So it is very difficult to compare to 10, 12. So so contamination should be avoided from the crime scene itself. So here you can see the more contaminated evidence, you can see the data. It's very difficult to analyze the sample. There is, you can see the minor and major profiles you can see in this one. It's very difficult for the the identification. So in STR data analysis, it's very important because there are a lot of artifacts, and the properly, and take time to analyze the samples one by one. Each locus should be identified proper. So alleles call here you can see 11, 13, 16. So there will be stochastic threshold because it depends upon the each DNA lab standard operating procedures. There will be highest chances of a stutter is there, pull up will be there, and many alleles will be dropout is there, imbalance is there. A lot of artifacts which you can see while you doing the DNA interpretation. So in the conclusion, if you are getting a clean profile, you can say this is evidence sample with the suspect included, source or contributor. So this questioned sample, which the suspect A is the contributor for the sample or the source of the sample, or you can exonerate or exclude. Then the third is inconclusive. How if it is a mixture sample, contaminated sample, it is inconclusive. Sometimes we will not get enough data, so insufficient data. These are the DNA conclusion which eventually come. So why the crime from the crime scene, it should start protect the samples. If you are getting a good sample for the analysis, for the extraction, then only you can expect good DNA result. If the lab is accredited 17025, it will not help you to get a good DNA result. Your crime scene processing also should be having much quality there. So now there are a lot of new technologies. Next generation sequencing is there. So you might know about it. So this is more faster, or you'll get more data from biological samples. For example, evidence item here. I think my time is about to finish. So 10, 12, then you can infer 10, 12. So unknown sample. So there is no suspect, or there is no reference sample, or there is no data for comparison. So the sample remains unknown. So here you can see with the use of this next generation sequencing technology, you can get the ancestry, the phenotypical data, hair color, eye color, and skin color, which can help the investigation agencies to narrow down the suspects list. For example, this is the sample which I worked on it about the ancestry. So you can see which is a sample which is collected from a Mongolian Taiwanese friend. So his ancestry in this region. You can see another Sudanese friend. So it can the ancestry from Sudan. So this is another from Bangladesh. So these are the possible. The ancestry identification is possible, and also genetic genealogy is now very good technology which is using in US. Phenotyping, ancestry, kinship analysis. You can get phenotypical snapshots. Prediction analysis also nowadays possible. So there are technologies are out there and which is developing, and then after that, so nowadays in the COVID-19 lockdown, so how to present in the evidence in the court? Now the virtual courts are coming. So how you present from the crime scene? You have to have graphics, digital construction often used in courtroom to help judge to understand. So what you have done in the crime scene, how the evidence, processed properly, how the evidence collected. That's why the judge, which is, he is not a scientific expert. Unfortunately, the irony is that the scientist doesn't know law and the lawyer doesn't know science. So in that case, so you have to present everything as a scientific expert. You have to present properly with the supporting data. So you can see in this image, it's a falling case. So with the support of this graphics, where the injury, how he fell down, everything can be properly explain to the judge, get the maximum punishment or the conviction, whether it is to prove out there is accident or it's an homicide. So, see, you can see that when the forensic pathologist is doing postmortem, so the point of impact, there is an injury is there, which is a trauma is there, he can identify that. So this kind of technology will be useful in future. And virtual autopsy, I don't want to spend time. This you know about it. So, and DNA data bank. So India also planning to have DNA data bank, solving a crime and solving it quickly and preventing additional crimes by the same perpetrator. An offender who is not apprehended and in a timely manner remains free, and he will commit the crime. So if data is there, so the police can easily catch the culprit or the perpetrator. So the very important DNA data bank. So many countries already they have national data bank. India also passed, I think, the DNA data bank bill in Parliament. It is now they are going to submit in the Rajya Sabha. Yes, that I know. And another challenge which I have noticed that the paternity testing samples nowadays, a lot of infertility clinics are there. There are many sperm donors, and the authentic reference sample. How we know that there is a mass disaster victim identification case? The biological, the legally the child belongs to the legally married couple, but they got this baby from an assisted reproductive technology from unknown sperm donor. So this is also another key area to be addressed. There are a lot of social and legal complexities in parentage determination in this case. If it is not, the data is not properly presented for the DNA analysis, because a DNA analyst doesn't know. He is only checking the biological connection or the biological kinship analysis. But it is from unknown sperm donor. The actual father is not the father. It's a legal father, but not a biological father. So a lot of paternity, or the assisted reproductive infertility clinics, which is happening in India. So there will be a lot of challenges in that case. I do not want to stress much on that. So what is generally happening? The forensic lab must be accredited, ISO IEC 17025, quality manual referenced each section, environmentally controlled laboratory with secure access, designated head laboratory responsible for its operation, having complete understanding of its measurement, procedures, and operation of its equipment, quality management system, QMS should be in place, quality management system in place. Appropriate range of reference standards, each with traceable calibration, suitable range of test and measuring equipment, a series of standard operation. So there are a lot of benefits for accreditation: establish quality in testing, the reliability, prevent risk, detect deviations, correct errors, improve efficiency, and ensure data quality and integrity. So it's time for transformation. Embracing change, forensic and allied science has no longer been just a science related to law and criminals. It has also entered in day-to-day life. The common man. Lawyers do not understand science, and scientists doesn't understand law. Forensic science becomes more and more complex, resolving forensics-based disputes in the courtroom becoming increasingly difficult. Fairness and administration of justice is the hallmark of a democratic and progressive society. So there are many missing links. So in order to improve that the young generation has to do a lot. So, and that DNA Innocence Project in US, there is a project called DNA Innocence Project. 350 exonerations wrongly convicted based upon DNA results. And he is the head of the project, Professor Hamikin, and he's a good friend of mine as well. So there are a lot of exoneration. So the US, they have a good laboratory systems, DNA databasing system. Even then, 350 exoneration. That means wrongly convicted and sentenced to jail with the DNA results. So then, what about in our country? We need good standard practices in DNA examination. So one of the things, training and education, in order for a qualified forensic science experts to testify competently about the forensic evidence, they must be first find the evidence in a usable state properly. This is the training which I did in Kerala, because I demonstrated how to do proper crime scene investigation. So let's together for a smart CSI and forensics for the future. And I think I exceeded my time, allotted time, and thank you very much. If you have any doubts, feel free to contact me, and I hand over the platform to the host. Thank you.
Thank you very much, sir, for the very informative session. Now, moving into the other speaker, we welcome Ms. Francisca Ventisca, member of Uni Bio for Association, Union of Italian Forensic Biologists, Forensic Biology Trainee at Forensic.India. Ma'am, please.
Good morning, everyone. I'm very glad to participate as speaker in this conference about crime scene investigation and forensic DNA typing. I would like to thank Forensic.India for giving me the opportunity to collaborate together. I really enjoyed all the activities and events where I can make a little contribution. Today, I'll show you an introduction of forensic science, and then I'll focus on an interesting research about the application of forensic methods to central blood meal analysis to identify the infection reservoir of anthroponotic visceral leishmaniasis. Let's start with the introduction to forensic science. The forensic DNA is the genetic material extracted from biological samples, such as DNA content in a drop of coagulated blood, semen, or in the few epithelial cells that can be found in a cigarette butt. In this case, it's called DNA from biological evidence. Forensic genetics is one of the disciplines of forensic science, and it's applied in modern cases in order to satisfy the legal and the human rights. In forensic genetics, the increase in sensitivity in technologies for DNA analysis, such as, for example, improvements in the ability to analyze minute amounts of DNA, made it possible to analyze a broader spectrum of materials found at the crime scenes, and also allowed so-called cold cases, that are the old unsolved cases, to be reopened and solved. Now let's focus on the human identification history in brief. In 1879, Alphonse Bertillon devised the first scientific system of personal identification using body measurements known as anthropometry. He claimed each person has different body proportions. Thus, body measurements and photographs were enough for individual identification. This method was used for 30 years, but it often failed. And in this picture, you can see the procedure of this method. In 1900, fingerprints were adopted as a recognition method. Fingerprints have been successfully used for identification purposes for more than a century and remained one of the most commonly recovered evidence at the crime scene. Due to their uniqueness and individualistic characteristics, the fingerprints are considered as the most powerful and widely used biometric characteristics. Then, a simple procedure consisting in determining the blood type AB0 and AB of a dried blood stain for human identification was discovered, but it was not reliable. The AB0 blood group system, which was the first genetic tool used for distinguishing between the individuals, can be performed in a few minutes, but it's not very informative. There are only 4 groups that are typed A, B, AB, and 0, and 40% of the population is type 0. Thus, while the AB0 blood groups are useful for excluding an individual from being the source of a crime scene sample, the test is not very useful when an inclusion has been made. especially if the sample is type 0. In 1985, DNA fingerprints or DNA typing, that is commonly called profiling, was discovered by the English geneticist Alec Jeffreys. Actually, he found that certain regions of DNA contain DNA sequences that were repeated over and over again next to each other. He also discovered that the number of repeated sections present in a sample could differ from individual to individual. By developing a technique to examine the length variation of these DNA repeat sequences, he created the ability to perform human identity tests. These DNA repeat regions became known as variable number of tandem repeats, which stands for variable number of tandem repeats. The technique used by Jeffreys to examine these sequences was called restriction fragment length polymorphism, because it involved the use of a restriction enzyme to cut the regions of DNA surrounding the variable number of tandem repeats. But there are some disadvantages. Actually, variable number of tandem repeats are a type of mini-satellite DNA in which the repeated sequence is generally from 10 to 100 base pair, and the number of times the sequence repeats varies among individuals. They are used for identification purposes, but not applicable to highly degraded DNA, since successful profiling requires large amounts, it means about 400-1000 base pair of intact DNA. This procedure is also expensive and not rapid. So then, in 1983, Carey Mullis developed the polymerase chain reaction that is now a common and often indispensable technique used in medical and biological research labs for a variety of applications, such as, for example, the short tandem repeats analysis that we will see later for the research. Here you can see the difference between the variable number of tandem repeats and the short tandem repeats. The first were replaced by the short tandem repeats due to their smaller unit size that is composed by from 2 to 6 base pair instead of the repeat unit size of variable number of tandem repeats that is composed by hundreds base pairs. So the DNA fingerprint is restricted to the detection of microsatellites. These are also called short tandem repeats, in which the repeated sequence is generally from 2 to 6 base pair, and the number of times the repeat occurs is variable among individuals. They are used for identification purposes, and they are also the better candidates for highly degraded DNA typing, since successful profiling requires less amounts, that means 100-400 base pair of DNA. Let's see the purposes of forensic DNA analysis. So why we use a forensic DNA analysis? First of all, to establish paternity and other family relationships, for example. Then for forensic examinations, such as to identify the potential suspects whose DNA may match evidence left at the crime scenes, to exonerate a person wrongly accused of crimes, and then to trace analysis and comparison with suspicious donors, such as victims, suspects, witnesses, investigators, and so on. Also, in cases of sexually assault, hypothetical reconstruction of the dynamics of affect. For identification in mass disasters, identification of missing people, construction of DNA database. And the importance of DNA analysis for individual identification was further demonstrated in the aftermath of the World Trade Center attack in 2001 and the tsunami catastrophe in 2004. Now let's see this event in detail. I think you remember the World Trade Center that occurred in September in 2001. In this picture, you can see the situation before and after this attack. Well, a great amount of debris, heat, and microbial decomposition of remains from about 3,000 individuals were there. Obviously, new strategies would be necessary to quickly prepare and organize DNA profiles and compare them with DNA profiles from relatives. DNA techniques were used to identify the remains of victims. Within 24 hours, collection points had been established around the city. Family filed missing person reports and provided buccal swab for DNA isolation. Personal items from the missing were also collected. And also several companies were involved in developing new software programs to help match DNA samples from family members to DNA profiles of victims. In this case, short tandem repeats, mitochondrial DNA, and single nucleotide polymorphism analysis have been done. Another tragic event that I think you remember, it was the South Asian tsunami that occurred in December 2004, where about 225,000 deaths were present. In this case, mitochondrial DNA and Y short tandem repeat markers were used to trace the maternal and the paternal inheritance, respectively, in order to identify the kinship relationships. In this case, within 3 months, 800 victims have been identified. Now let's focus on the research entitled "The Potential Use of Forensic DNA Methods Applied to Sand-Fly Blood Meal Analysis to Identify the Infection Reservoir of Anthroponotic Visual Leishmaniasis." These pictures show this disease, the consequences. But before introducing this research, I want you to know a basic knowledge about parasitology. This is the science that deals with the organisms that take up their abode temporarily or permanently for the purpose of procuring food and the relationship of this organism to the host. It's a type of symbiosis where any plant, animal, or protist is intimately associated with another organism of the same or different species. There are various types of symbiosis: mutualism, commensalism, and parasitism. What are the different kinds of symbiosis? The mutualism is the condition where both organisms benefit. The commensalism is the conditions where one organism benefits and one organism is unaffected. And parasitism is the condition where one organism benefits and one organism is harmed. These are the parasites, and the hosts are the protagonists of this discipline. Parasites are organisms needed another organism called host to survive and can be facultative if it's able to live both free living and parasite living, obligate living permanently in a host and cannot live without a host, and spurious free living parasites passing through alimentary tract without infecting the host. Then hosts can be definitive, in which parasites replicate sexually; intermediate, that is also called vector, in which the parasite is metabolically dependent to complete its life cycle and undergoes essential development; accidental, a host that the parasite does not normally infect may or may not complete development; parathenic, a transport host, but the parasite does not require it to complete its life cycle, hence thus the reservoir, a host that maintains a parasite population and has the potential to transmit to a human or domestic animal. It shows no sign or symptoms of disease. Now let's see what is the visceral leishmaniasis. From these pictures, you can see the Leishmania donovani parasites, as shown in the small picture. These are demonstrated in buffy coat preparations of blood and bone marrow aspirates. In the Indian subcontinent, this parasite is transmitted between people by the sandfly Phlebotomus argentifes, as shown in the picture below. Visceral leishmaniasis is also known as Kala-Azar and is a fatal form of leishmaniasis caused by the parasitic protozoan Leishmania donovani and transmitted by the sandfly Phlebotomus argentifes. This can often be fatal if not treated properly. In a time period ranging anywhere from a few months to a few years since the bite, an infected person can start exhibiting symptoms such as fever, weight loss, reduction in blood iron levels, and damage to internal organs like liver, bone marrow, and spleen. What about epidemiology? Leishmania, there are different kinds of types of Leishmania, such as infantum, that is diffused in the Mediterranean basin, North Africa, and Central and South America, where the infection reservoir are animals, in particular dogs. And then there is the Leishmania donovani in the Indian subcontinent, where it has an anthroponotic transmission cycle. There are 4 different groups of infected people that can carry the parasites. Patients with active disease, cured visceral leishmaniasis cases, patients with post-Kala-Azar dermal leishmaniasis, and asymptomatic individuals. Now let's see something about numerical data. These data are reported in the World Health Organization website. If you are interested, you can search all kind of information here. The picture shows a young girl affected by leishmaniasis in Pakistan. So about 300,000 new cases occur every year, with mortality rates of about 10%. According to the World Health Organization, in India alone, around 10,000 cases were reported in 2014. Given the high rate of non-reported cases, the actual number is estimated to around 100,000, the majority of whom live in poor rural settings in the northeast state of Bihar. What are the symptoms of visceral leishmaniasis? First of all, the enlargement of the spleen and enlargement of the liver, as you can see in this picture, the night sweats, the severe temperature or irregular bouts of fever that can last for weeks, bleeding, blackening of the skin, scaly skin, darker and harsher skin, cold, weakness, and also weight loss. Now let's see the cycle of these parasites. This is the anthroponotic transmission cycle. The disease is transmitted by the bite of the female sandfly that requires a blood meal for egg production. Various ecological parameters, including air temperature, rainfall, wind speed, relative humidity, soil moisture, pH, and organic carbon, are known to influence the oviposition of female sandflies, as well as the survival and the development of larvae. Let's see in detail this cycle. First of all, there must be a sandfly that takes a blood meal. It injects the promastigot stage into the skin. Then promastigots are phagocytized by macrophages or other types of phagocytic cells. Then promastigots transform into amastigots. The amastigots multiply in cells of various tissues and infect other cells. Then the sandfly takes a blood meal again. It ingests the macrophages infected with amastigots. It ingests the cell with parasites. The amastigots transform into promastigot stage in the gut, and they divide in the gut and migrate to the proboscis. So again, the sandfly that takes a blood meal on a human injects the promastigot stage into the skin, and the cycle starts again. What is the methodology used in this research? Forensic DNA methods recover human DNA profiles from the blood meals of infected sandflies to directly trace the human source. Human DNA profiles are matched to reference DNA samples from individuals living or working where the sandflies have been collected. Human DNA profiles are generated within 24 hours after blood feeding. Nested PCR method is used to detect parasites present in a fresh 24 hours blood meal in flies fed on infected hamsters. What about materials and the methods? Forensic DNA methods have been applied to extract human DNA from blood-fed Leishmania donovani infected flies. These methods involve the analysis of highly polymorphic short-tandem repeats markers. Forensic short-tandem repeats kits allow the generation of DNA profiles and can type multiple highly polymorphic short-tandem repeats in a single reaction, producing profiles that are unique in a population and that can be matched to a reference database for source identification. What are short-tandem repeats? As I told before, they are the markers used in this research. They are repetitive sequence elements composed from 2 to 6 base pairing length. The total size of a short-tandem repeat is usually less than 500 bases. There are over 1 million sites called loci in the human genome that have short tandem repeats, of which over 10,000 have been characterized. The more loci that are amplified, the higher the statistical power of discrimination, obviously. And it's the best solution for human identification in forensic analysis, including high power of discrimination and rapid analysis speed. From this image, you can see a representative figure of the short tandem repeats. A person inherits a copy that is called the allele of the short tandem repeats from each parent. So one from the mother, one from the father. And in case of homozygote condition, both alleles are the same length. In case of heterozygote conditions, alleles differ and can be resolved from one another. Let's see the data. Forensic DNA methods have been applied to recover human DNA profiles from the blood meals of infected sandflies that have been matched to reference DNA samples from 2 volunteer individuals living or working in the vicinity of the sandfly collections. We found that the ability to obtain readable human DNA fingerprints from sandflies depended entirely on the size of the blood meal and the kinetics of its digestion that we will see later. So from this table, you can see the profile, the successful profiles obtained. The profiles with the 2 plus means the profiles was full because both alleles of heterozygous loci are readable, as we can see in the first lines. Where you see just 1 plus, it means that it is a partial profile where only heterozygous or homozygous allele is readable. When you don't see the plus but you see a minus, it means no profile. None of the alleles are detectable. So, what about the results? With this table, we want to show the quantity of human DNA in flies is proportional to blood meal size. We found that the ability to obtain readable human DNA fingerprints from sandflies depended also on the kinetics of its digestion, as I told before. From these pictures, you can see the kinetics of blood meal digestions. The picture A shows that flies with a full or partial blood meal at day 0, and the kinetics of blood meal digestion on day 1, 2, and 3 post-blood meal. From the picture B, you can see the total and human DNA at days 0, 1, 2, 3, and 5 post-blood meal. You can see the decline, and this is confirmed by the picture C, where the percentage of flies with a detectable short tandem repeat profiles. It declines from day 0 to day 5. What about the detection of these parasites in fresh blood meals from Phlebotomus argentifes? Leishmania donovani parasites have been injected into hamsters. After 2 months, when they had lost 30% of their weight, they were anesthetized, their abdomen were shaved, and they were exposed to 100-150 Phlebotomus argentifes flies for 1 hour at 26 degrees in the dark. Non-fed females were removed from the cage, and the blood-fed flies were carbonic dioxide anesthetized and processed for DNA analysis. A highly sensitive nested PCR method is used in order to increase the assay sensitivity. And the target gene was NAD dehydrogenase subunit 5, which is located on the maxi-circle DNA and used for the Leishmania genotyping. Standard direct PCR, as you can see from the picture A, or nested PCR, that is the more sensitive method, were used. You can see this reaction from these pictures, and the DNA extracted from individual blood-fed flies fed on hamsters and mixed with the indicated number of parasites. These pictures just show the sensitivity of the higher sensitivity of the nested PCR compared to the classic, the normal PCR method. Here, 2 independent experiments, A and B, showing nested PCR amplification of the gene from flies fed on infected or naive hamsters. You can see here the arrows indicate flies showing positive reactions. Obviously, the performance in both experiments was higher in infected hamsters instead or naive. So, what about conclusions? The forensic DNA methods have been successfully applied to epidemiological studies of human visceral leishmaniasis and point out some of the limitations of this approach. The ability to obtain human DNA profiles depends on the size of the original blood meal and the kinetics of its digestion. This is attributed to the excretion of the blood meal remnants from the gut. The first 24 hours post-blood meal, the flies carry enough blood to obtain a human profile suitable for comparison. It becomes more difficult after 48 hours or longer. Another issue associated with the short time frame in which short tandem repeats profiling is effective is the potentially low number of the parasites present during the 24 hours after the fly has acquired an infected blood meal, probably because parasites are more concentrated in the skin respected to the bloodstream. To overcome this issue, a nested PCR targeting maxi-circle DNA was developed. A nested PCR targeting ribosomal DNA has been previously shown to substantially enhance the sensitivity and the specificity of Leishmania detection in the skin compared to the conventional techniques. This method is compatible with detecting Leishmania donovani within a fresh 24 hours blood milling flies fed on infected hamsters. So, this study demonstrates for the first time that the use of forensic DNA methods enables the identification of the human source of a sandfly blood meal and may therefore be used to directly trace the source of an infected blood meal in flies recovered from Kala-Azar endemic zones. This happens because sandflies are weak flyers and travel in short hops rather than in sustained flight. There is a high likelihood that their human blood meal source will be found living or working within close proximity to the location of the capture. Understanding the dynamics and epidemiology of anthropogenic transmissions holds clear importance for the development of control strategies for human leishmaniasis. Here is my social media LinkedIn if you want to ask me something about this issue or something else, and my email if you want to contact with it. Thank you for your attention.
Thank you, ma'am, for the valuable session. Now, moving on to the other speaker, I welcome Dr. Anna Barbaro. She is PhD in Forensic Genetics, President Worldwide Association of Women Forensic Expert, and also study medical and forensic investigation in Italy. Ma'am.
Hi, good day. Thank you very much for the invitation to this conference. I'm going to share my video. Please allow me to share my presentation. Okay. I'm going to talk about crime scene investigation and forensic DNA typing. Forensic science is the application of science to the resolution of legal issues. My laboratory, CMF, is placed in the south of Italy, in Reggio Calabria. That is a region where many forensic exports happens due to the mafia problem. My laboratory has several departments in which we are able to perform different kinds of criminalistics analysis, such as ballistics, forensic biology, forensic genetics, underwriting analysis, latent prints examination, digital forensics, legal medicine, ballistic and electronic microscopy, toxicology. We have also a CSI unit able to go directly to the crime scene for collecting samples. Forensic science is the application of science to the resolution of legal issues according to the definition of the American Academy of Forensic Science. And because of this, forensic science includes criminalistics and criminology. Criminalistics consists in the application of scientific methods to the recognition, collection, identification, and comparison of physical evidence found at the crime scene. Hans Gross is considered the father of criminalistics. Criminology is different from criminalistics, even if sometimes there is a big confusion between criminalistics and criminology. Criminology is the study of crime and deviant behavior. Criminology is an interdisciplinary field in both the behavioral and social science. The Italian Cesare Lombroso is considered the father of criminology. As well known, forensic science starts at the crime scene. This because, according to Locar's exchange principle, every contact leaves a trace. There is a clear relationship between the elements at the crime scene, between the victim, the suspect, the items found at the crime scene. Because of this, the crime scene contains information that must be in a systematic, legal, scientific way for the collection of the evidences that can be used as a proof to rebuild the sequence of the events. The aim of a crime scene investigation is to answer to some questions, such as what happened, where happened, when, how, who was the victim, who was the killer, to whom the attack has been performed. There are several types of crime scene. Here you can see an example of crime scenes from real case works of our laboratory. A closed crime scene, an open crime scene, mixed crime scene, and sometimes it's possible to observe underwater crime scene. It's possible to classify different types of crime scene. Primary crime scene is the place where there is more contact between the aggressor and the victim, where more time is spent and where the greatest number of attacks is carried out on the victim. So it's the most important scene for searching forensic evidences. Often, but not always, it's the scene where the victim body is found. Secondary scene is the scene where the interaction between the aggressor and the victim is lesser in comparison to the primary one. It may be the scene where the body is found. The intermediate scene is the scene which generally serves to move the body from the primary scene to the one where the body is left and then found. For example, it may be a car, a train, or something like it. There are some fundamental rules that must be followed during the investigation of a crime scene. For example, the preservation of the state of the scene in order to avoid evidence alteration. Evaluation of time and environmental conditions. General description of the place. Planimetric study using sketch, photo, video, 3D laser reconstruction. Description of each element at the crime scene, measurement, right to collect evidence, packaging and labeling. Adequate evidence storing conditions, accurate chain of custody, and some precautions to protect such as wearing gloves or ties. This may be resumed in three words: observation, registration, and preservation. Because of this, crime scene investigation is a technical activity that must be carried out by technical staff with specific knowledge. An incomplete inspection of the scene in most cases affects the result of the investigation. If errors are done within the first 24 or 48 hours, and they can compromise case resolution. In our experience, we have seen that errors performed at the crime scene were compromised on the investigation, and the crime difficilely was solved. One problem is concerning crime scene staging. Staging consists in the actions taken by the offender for altering the crime scene in order to deviate the investigation. The most common type of staging occurs when the author changes elements of the scene to simulate a suicide or accident. The most common type is when the author tries to redirect the investigation, simulating sexual homicide. At the crime scene, it's possible to find biological or not biological evidences. Biological evidences may be evident, such as blood stains, or latent. Here you can see some images taken from our real caseworks. In case of latent evidences, they must be searched by special forensic lamps, or in case of latent blood, it may be searched using chemical reagents such as luminol. Obviously, don't forget to classify and photograph evidences using meter reference before their collection. Another important issue is concerning contamination. It may be caused by investigators at the crime scene or by error of laboratory technical staff, or due to bad collection preservation procedures. It may occur between persons and evidences or among evidences themselves. Because of this, in order to minimize contamination, there are some precautions, such as wearing gloves and changing them, using a mask, using a single-use material, analyzing evidences at separate times from the reference samples, properly storing samples. Because of the complexity of crime scene investigation, it must be performed by qualified and trained personnel, following the international procedures and protocols that have been developed for the right crime scene evidence collection and investigation. Only if adequately collected and preserved, the evidences may be useful for further analysis. As I told before, an incorrect CSI may compromise further investigation. The ISO 17020 has been developed for the certification of people who performs crime scene investigation. It's an international protocol. Samples taken at crime scene must be adequately transferred to the criminalistic laboratory. The laboratory is performed the analysis of biological or not biological samples collected at the crime scene in order to identify them, or in case of biological evidences, in order to define the type of biological material before to perform DNA typing. DNA is found on 2 chromosomes. Each person receives half of the genetic information from the father and the other half from the mother. Because of this, with the exception of identical twins, each individual genetic profile is unique, such as fingerprints. The genetic profile is the same in all the tissues of an individual. This means in saliva, blood, hairs, bones. DNA is useful in paternity tests and criminal investigations. Paternity tests may be performed as a prenatal test, postnatal, or post-mortem test. In addition, it's possible to perform maternity tests, siblings analysis, sisterhood analysis, or reconstruction of complex family trees. In the criminal investigation, DNA is used to identify a victim, for example, from a part of the body, such as it happens during DWI investigations, or to identify a suspect on the basis of the evidences found at the crime scene, or to link different crimes. Because of this, national DNA databases have been developed all over the world. At the crime scene, DNA is virtually anywhere. The victim and the aggressor leave their DNA wherever they have been. DNA typing consists in the detection of variations in the number of tandem repetitions of some sequences. STR, short tandem repeats, are the markers we generally analyze. The number of variation is different from individual to individual. This variability is called polymorphism. This allows to obtain a genetic profile. STRs have been found on autosomal and sexual chromosomes, which are placed in the nucleus of the cells. STRs became rapidly the first choice markers for human identification because of their features and the good polymorphism degree. Another type of DNA is the one placed in the mitochondria of cells. It's inherited through the female lineage and it's used for degraded DNA analysis. This is an example of a DNA laboratory organization with separation between different rooms: extraction, quantification, pre-amplification room, and then amplification and sequencing room. Obviously, the area dedicated to the sample screening must be separated from the other areas of the laboratory. Here you can see an example of HID workflow. As you can see, DNA analysis is a multi-step process, it's a complex process, because there are several steps starting from DNA extraction to quantification, amplification, and detection. In 1990s, several SCR loci were selected from the forensic community for forensic applications. Once they have been definitively established, some commercial multiplexes were developed, and nowadays they are widely used either in forensic than in paternity tests because of their easy use and high discrimination power. Initially, multiplexes were simple and they included only few loci, while nowadays are available large multiplexes that allow the simultaneous amplification of many markers till 244. In fact, initially, 12 loci have been selected for the USA database called CODIS, but during years, new loci have been added following to the diffusion of databases all over the world, and especially after the Treaty of Prüm, signed by European countries with the aim of a cross-border cooperation and exchange of DNA profiles. The addition was due to the aim to reduce false matches, but also to increase international compatibility and discrimination power in case of degraded samples or missing persons identifications. There are now large multiplexes that allow simultaneous amplification of European and USA database core loci. Here you can see an example of a DNA profile obtained from a skull found in the sea after 6 months. You can see here's a DNA profile. This is another example of DNA profile from latent blood stains detected by luminol on the pants of a suspect. this person was suspected to have killed a woman. This is another example of DNA profile from anonymous letter with latent prints enhanced by ninidirin. These are real case works from our laboratory. When a comparison is done between DNA profile from DNA evidence and DNA profile from a suspect, there are two possible results. An exclusion, this means DNA profile from the evidence is different from DNA profile from the suspect. Or an inclusion, this means there is a compatibility between the two profiles. In this case, a biostatistical calculation must be done in order to evaluate what is the probability that the DNA found at the crime scene effectively belongs to the suspect in comparison with the probability that is from a random individual in the population that who has the same genetic profile of the suspect. Random match probability represents the possibility that the profile of a random person in a population matches the DNA profile of the suspect. The calculation of random match probability is using special forensic softwares and using population frequencies. Because of this, some studies are necessary to know the allele frequencies of the markers examined and to calculate how often a given genotype is found in the population. The likelihood ratio is a relationship between two probabilities of the same evidence under two mutually exclusive hypotheses. The hypothesis that the suspect is the donor of the DNA profile, this is called hypothesis of the prosecutor, as opposed to the hypothesis that a random unknown person in the population not related with the suspect is the real donor. This is called defense hypothesis. An interpretation table of likelihood ratio results has been developed by Hivet. In the last years, a change from the traditional laboratory to the mobile laboratory occurred. Because of these new instruments have been developed, Rapid ID system is a fast and simple to use instrument that produces DNA profiles in 90 minutes. The system integrates sample preparation and capillary electrophoresis in order to generate DNA profiles using cartridges where are placed all consumables. Initially developed for rapid tests of oral swabs taken to suspects at USA police stations, now it's useful also for the analysis of other kind of forensic samples. But what happens when, during a comparison, no match is found between the suspect and DNA profiles in the database, or when there isn't any suspect, there are no additional information in the investigation, there are no witnesses. In the last years, a new kind of DNA investigation has been developed, the so-called DNA investigative intelligence. This kind of investigation uses other markers called SNPs. SNPs are variations of a single base in the DNA sequence. They differ between different individuals of the same species or between pairs of chromosomes in an individual. SNPs are useful, for example, for the geographical ancestry prediction. They were used in 2004 in order to identify a terrorist during Madrid attacks. This snippet was prepared in the Santiago de Compostela laboratory where I was at that time. In addition, SNPs are useful for the prediction of physical features. For example, a system called IrisPlex has been developed in 2008 by Erasmus University diversity, and it allows sensitive and accurate predictions of hair and skin color. Future projects are going on in order to be able to perform age prediction, facial morphology prediction, height prediction, in order to differentiate between monozygotic twins. A consortium called BSAGE has been established with the aim to provide scientific knowledge and prototype tools for predicting appearance, age, and ancestry from DNA traces found at crime scene with investigative purposes. This is possible now because a new technology has been developed called next generation sequencing. It is a technology which has revolutionized genomic research, allowing to sequence the entire human genome within a single day. It's useful in forensic because of the efficient sequencing, the lower cost of analysis in comparison with traditional procedures, the high sensitivity, and the high multiplexing capacity. It allows, for example, to analyze simultaneously autosomal STRs, sexual STRs, SNPs, and mitochondrial DNA. But when DNA analysis, DNA results are admissible at the court, results of a forensic analysis may contribute to establishing the guilt or innocence of a person. So, forensic DNA typing requires great caution, responsibility, and professionalism. In fact, nowadays, modern technologies enable scientists to find and analyze minute pieces of evidence from a crime scene or indirectly connected with the crime. This enables to obtain information from samples that were previously not analyzable, and it allows solving cold cases. Because of this, several associations worldwide work together in order to develop protocols and to favorite the standardizations of methods. Quality certification is required to all forensic laboratories, and especially to the ones who want to submit their DNA profiles to the national databases. In this case, the ISO 17025 certification is required. Because of the complexity of forensic science and the many disciplines involved, this part requires a great competency that may only be achieved by specific education and training, pluriannual experience, application of standardized scientific protocols, participation in proficiency testing, and obviously stringent professional ethics. In addition, in some countries such as USA, this quality certification of the forensic experts is required according to ISO 17024. This is required in order to demonstrate the effective competence of that person to carry out the activity in a specific area and to discuss results at the court. In the last years, I have published some manuals about forensic procedures. For example, here you can see the manual forensic science that has been published in 2017 by CRC Press in United States, and the manual of criminalistics and criminology that is now in publication in Spain, in Spanish language. These manuals are the result of an international collaboration with many famous forensic experts in different forensic fields. If you have any questions, please contact me directly to my email. You can see here in the slide. And I invite you all to follow our social networks pages, my personal ones and the one of my association, the Worldwide Association of Women Forensic Experts. Thank you very much for the attention, and thank you again to the organizer for inviting me to this conference. Thank you.
Thank you very much, ma'am. From the behalf of Forensic.India, we thank all the speakers for giving their valuable time and sharing their knowledge with us. Also thankful to all the participants for attending the conference. Thank you.